بسم الله الرحمن الرحيم. السلام عليكم ورحمة الله وبركاته. أعزائي طلبة السنة الثانية، المحاضرة اليوم راح تكون Reproductive System Embryology. Objectives: Number 1. Describe the development of the gonads in male and females. Number 2. Understand the differences in male-female internal duct development. Number 3. Know the origins of the external genitalia. Number 4. Understand the developmental abnormalities in male and female development. Development of genital system. The genital system consists of gonads or primitive sex cord. Number 2. Genital ducts. Number 3. External genitalia. All the three components, the gonads, the genital duct, and external genitalia of the genital system initially pass through an indifferent period of development, which is the same in both male and female embryos. The differentiation of the sexual dimorphism depends on the SRY gene. It is a sex-determining region on the Y, localized on the short arm of the Y chromosomes. Look here, this is the Y chromosomes. This is the short arm of Y chromosomes. This is the long arm of Y chromosomes. In the short arm of my Y chromosomes, there are sex-determining region Y, SRY gene, and this is present only on the Y chromosome. It is absent from X chromosome. Now, the gonads pass at the beginning in the indifferent stage of gonadal development. It is the same in both male and the female embryo. The gonads are formed by proliferation of the coelomic epithelium and a condensation of the underlying mesenchyme, forming the genital or gonadal ridges. Now look here, this is a 4 weeks embryo. Now the 4 weeks embryo, the gonads are indifferent. That is to say, there is formation of the genital ridges or gonadal ridges. This is formed due to proliferation of the coelomic epithelium and condensation of the underlying mesenchyme. So the genital ridge or gonadal ridge is seen in both male and female embryos. The primordial germ cell start to appear in the lateral wall of the yolk sac, and these primordial germ cells migrate, and it will pass by amoeboid movement and invade the genital ridge during the 6th week of development. If they fail to reach the ridges, the gonad do not develop. So the primordial germ cells first appears in the wall of in the lateral wall of the yolk sac. It migrates by amoeboid movement to reach the genital ridges during the 6th week. Now, when the primordial germ cells reach the genital ridges, the epithelium start to proliferate and forming irregular cord, and this irregular cord is called a primitive sex cord, and this primitive sex cord remain in close contact with the surface epithelium. So just to remind you what happening during this stage: number 1, formation of the genital ridge or gonadal ridges, number 1. Number 2, the primordial germ cells migrate from the lateral wall of yolk sac and they reach the genital ridges. Number 3, there is a proliferation of coelomic epithelium and formation of a primitive sex cord. This primitive sex cord, they remain in close contact with the surface epithelium. The indifferent stage will be followed by stage of differentiation. Here there will be formation of testes or formation of the ovary. Under the influence of the Y chromosomes, the primitive sex cord will proliferate and start to penetrate deep into the medulla, and they will form the testis or medullary cord. Toward the hilum of the gland, the cord break up into a network of cell strands, and this will later give rise to tubules of the rete testis. During further development, there will be formation of a dense layer of fibrous connective tissue, and this layer is called the tunica albuginea. This will separate the testis cords from the surface epithelium. So, development of the testis is under the influence of Y chromosomes, and the primitive sex cord, which is formed in the indifferent stage, now proliferate and start to be penetrate more deep into the medulla, and it will form the medullary cord or testis cord. This medullary cord or testis cord start to degenerate in this area in the hilar region and will give rise to the rete testis cord. At the periphery, there are appearance of fibrous connective tissue. This will separate the testis cord from the surface epithelium. So 3 things. Number 1, appearance of the testis cord. Number 2, formation of the retitestis cord. Number 3, the tunica albogene. In the 4th month, the testis cord become horseshoe. This is first point, and become continuous with the retitestis. And there will be 3 types of cells. The first is called the primitive germ cells. The primitive germ cells is derived from the primordial germ cells, which is coming from the wall of yolk sac. The second is called sustentacular cells of Sertoli, and this will be derived from the surface epithelium of the gland. The third is called the interstitial cell of Leydig, and this is derived from the mesenchyme of the gonadal ridge, and it is found between the testis cord. Now, the interstitial cell of Leydig, which is formed from the mesenchyme, start to secrete testosterone and produce it by the 8th week of gestation, and so the testis become able to influence the sexual differentiation of the genital duct and external genitalia. Now, the testis cord will remain solid up to the puberty, and then it become canalized, and they form the seminiferous tubule. Now, these seminiferous tubules, they are connected to the tubule of the retitestis, and these tubule of retitestis will enter the ducta efferentia. Now, and this ducta efferentia connect the retitestis with the mesonephric duct or Wolffian duct, which will become the ductus deferens. Now, just to remind you that this is the seminiferous tubules, which will become canalized at the puberty. The primitive germ cells will become spermatogonia, and it will be supported by Sertoli cells, and there is interstitial cell of Leydig, which secrete the testosterone. Now, look here, you can see there are the retitestis cord, and if you just to remind you that this retitestis cord is actually, it is the medullary cord or testis cord, which start to degenerate toward the hilum and form the tubule of retitestis. Now, these retitestis, they are connected with the ducta efferentia or efferent ducts, from where this ducta efferentia come. Now, if you remember this drawing, which we have taken it in the urinary system, you remember that the second stage of the kidney development is called the mesonephros, which consists of mesonephric tubule and mesonephric or Wolffian duct. Most of the mesonephric tubule degenerate. Some of them which retain and remain, they will form ducta efferentia, and this ducta efferentia connect the retitestis with the mesonephric duct. The mesonephric duct or Wolffian duct will persist and will be changed to the ductus deferens. So we have these are the testis cord, which will form the seminiferous tubule. They are connected with the retitestis, and this will be connected with the ducta efferentia, and this will be connected with the vas deferens or ductus deferens. Cryptorchidism is one of the abnormality of the testes. Sometimes a doctor examined a baby and couldn't found one or both testes. It is absent from the scrotum, and this is due to the failure of the descent of the testis, and this testis may retain in the abdomen or may stop at deep inguinal ring or inguinal canal or in the superficial inguinal ring at root of the penis or perineum. The undescended testes fail to produce mature spermatozoa. Ectopic testes, the testes may take a position away from the normal pathway of the descent, as to be found in the lower part of the anterior abdominal wall or upper part of the thigh. Just to remind you that the testis is at the beginning is located in the abdomen. Due to the growth in the length and due to the increased intra-abdominal pressure because of the development of the organ, the testes start to descend and it will come down to be located in the scrotum. Development of the ovary. In the female embryo with an XX set of the chromosome, sex chromosome complement, and there is no Y chromosome, the primitive sex cord, which we speak about it in the indifferent stage now, will start to be broken into irregular cell clusters, and later they disappear and they are replaced by vascular stroma that form the ovarian medulla. So the primitive sex cord will be broken and it will form irregular cell cluster and disappear and replaced by vascular stroma, and this vascular stroma will give rise to the ovarian medulla. Now, the ovary. The surface epithelium of the female gonad will continue to proliferate, and in the 7th week it gives rise to a second generation of the cord, and these are called cortical cords. And these cortical cords, they remain into close contact to the surface. In the 4th month, these cords start to split into isolated cell clusters, and each one of these clusters formed of one or more primitive germ cells, and these primitive germ cells will subsequently later on develop into oogonia, and the surrounding epithelial cell will form the follicular cell. Together, the oogonia and the follicular cell will constitute the primordial follicle. So in the ovary, we have the cortical cord, and these cortical cord developed from the surface epithelium. We have degenerating medullary cord, and this degenerating medullary cord will give the ovarian stroma. The cortical cord will be split into isolated cell clusters, and these cell clusters, each one will be formed of primitive germ cells. And if we remember that this primitive germ cells is derived from the primordial germ cells, which is coming from the wall of yolk sac. But these primitive germ cells in the ovary will be subsequently developed into oogonia, and the surrounding cells which surround it will called follicular cell. The oogonia and the follicular cell which is surrounding it will give rise to the primordial follicle. So just to repeat what we have taken, we said that the gonad will pass into indifferent stage of the gonad, and later on there is a different stage of gonad. Now in the first, you remember that the sex of the baby is determined at time of fertilization. There is 44XY for the male and 44XX for the female. In the male, the Y influence affects the gonadal development, so there will be formation of the testes. In the testes, the medullary cord develop, and there will be no cortical cord, and there is 3 types of cell: the primitive germ cells, which will be changed to the spermatogonia, and the circulate cell, and the other cell which is called the interstitial cell of Leydig. And there will be formation of thick tunica albuginea. While in the ovary, due to the absence of the Y chromosomes, the medullary cord degenerates, and it will give rise to the ovarian stroma. The cortical cord develop, and this cortical cord will give cell cluster, and this will surround the primitive germ cells, which will be turned to the oogonia. The oogonia with the surrounding follicular cell will give the primordial follicle. There is no tunica albuginea, but there is very thin capsule surrounding the ovary. Genital ducts. Genital ducts, like the gonads, also there is indifferent stage of the genital ducts. That is to say, in both male and female embryo, there are more or less similar in the development, and both of them, they have initially 2 pairs of genital ducts that are called the Wolffian ducts, mesonephric, and the paramesonephric or Mullerian ducts. Now these 2 ducts, the paramesonephric, the Wolffian is present, and you remember it, it is coming from the mesonephros, from the which we discussed with the renal system. Now the paramesonephric duct arises as a longitudinal invagination of the epithelium on the anterolateral surface of the urogenital ridge. This paramesonephric duct have an expanded cranial end of the duct, and it is open into the abdominal cavity, while caudally it first runs lateral to the mesonephros, and later on it crosses it ventrally, and then it grows in caudomedial direction toward the midline, and where it becomes in close contact with the paramesonephric duct from the opposite side. Now just look to this drawing. You can see this A, it is a male, and B is a female. In the A, you can see here this is the testis, here the testis cord, the tunica albuginea, and the rete testis. Here in the ovary, you can see the cortical cord of the ovary, the degenerating medullary cord, and there is no tunica albuginea. Now there are 2 types of, although this is female and this is male, you can see in both 2 types of the ducts. One of them is called the mesonephric duct or Wolffian duct, and the other is called the paramesonephric or Mullerian duct. Here also you can see this is the male duct, Wolffian duct. This is the female duct, which is the Mullerian duct. Now the 2 ducts are initially separated by a septum. Now this is the Mullerian ducts come, and this is the other from the other side. They come to fuse in the middle, and then the two ducts, they are initially separated by a septum, but later both the left and right Mullerian duct, they will fuse and they will form the uterine canal. Now the caudal tip of the combined ducts, the right and left, will enter the cloaca, and after its divisions, the cloaca, the ducts now enter the urogenital sinus, and they will form a small elevations in the posterior wall of the urogenital sinus, and this is called a paramesonephric or Mullerian tubercle. Now look here, you can see here, this is a male and this is a female. Both of them, they have the mesonephric and paramesonephric duct. Now look to the paramesonephric duct. The paramesonephric duct has a cranial end and it is open into the abdominal cavity. It passes just lateral to the mesonephric and later across it ventrally, then caudomedially, and then it will come in close contact with the paramesonephric duct of the other side, and they fuse together, and they will open in the posterior wall of the urogenital sinus at the Mullerian tubercle. At the site of this Mullerian tubercle, there will be opening of the mesonephric or Wolffian ducts on each side. This is one and this is the other. So at this stage, again, this stage is called indifferent stage of the development of genital duct, because in male and female, both you can see both the Mullerian and the Wolffian ducts, the Wolffian and the Mullerian ducts, both of them, they are seen in the embryo. Now, differentiation of the duct system. Now, we say that there is mesonephros and paramesonephros, the Wolffian duct and the paramesonephros, the Mullerian ducts. These two ducts, we say that they are opening in the urogenital sinus. The growth and development of the genital ducts and the external genitalia will occur under the influence of hormones circulating in the fetal blood. Now, the hormones in the male, the fetal testis will secrete inducing substance and Mullerian inhibiting substance, which is called anti-Mullerian hormone, and this will induce the development of the mesonephric duct, or another name for it, Wolffian duct, and suppress the paramesonephric duct, which is the female duct system. So the testes also secrete androgen hormone, and this stimulates the development of prostate, seminal vesicle, penis, penile urethra, and fusion of the scrotal swelling. While in the female, due to the maternal and placental estrogen, number one, number two, absence of the male-inducing substance and androgen, the paramesonephric duct will start to develop and differentiate into uterine tube, uterus, cervix, and upper vagina. And estrogen, which is due from maternal and placental source, act on the external genitalia of at the indifferent stage, and it will form the labia majora, labia minora, clitoris, and lower vagina. Today we have taken the development of the genital system, we have taken the gonads, we speak about the indifferent stage of the gonad, and we speak about the formation and development of the testis, development of the ovary, and we start with the genital duct development. We start with the indifferent stage of genital duct development. Next lecture, we will speak about the development of the genital ducts in the male and in the female, and also we will discuss the external genitalia. Thank you for your listening. Keep study hard and have a nice day. Thank you again.